Good morning and welcome back to the class. So we finished about some of the applications of biology in the fields of medicine, and we have seen that biology as a science is quite important for our survival. Now in today's chapter we are going to start to understand how microorganisms are important to us. We might be wondering where microorganisms belong, because these are called as microorganisms. So the name suggests that they are very, very small. Now, what role do you think that it plays for our survival? How does it help us to survive? In fact, when we learned about biological diversity, we have seen that these microscopic organisms are present everywhere. You take soil, you take water, you take inside our body, you take outside our body, you take our surfaces, you take where uh, very high temperatures or very low temperatures, you can see that microorganisms are present everywhere. I can call them as omnipresent. Not only that, we also see that microorganisms are diverse. What do we mean by that? We have various types of microorganisms. We see small particles which are not even living organisms called as the viruses. Some of them which are smaller than viruses, we call them as viroids and prions. I'm sure you remember all this that we have learned in our last year. Along with that, we have various bacteria, we have fungi, we have protozoans, unicellular microorganisms, and we also see that they are all having certain protein structures which have the ability to cause some infections in our body. So you see that their sizes are very, very small, but the effect they induce on us is something which is too high. And because of all these effects, uh, you know many of them can be used for us. At the same time, some of them are causing diseases. And in our last chapter, we have seen that certain microorganisms cause diseases. We learned about few important diseases. We saw how it affects us, what are the important symptoms of those diseases, and so on and so forth. But today we will be focusing more on certain microorganisms which are very helpful for human welfare. Now, before I start off talking about human welfare, you should know that the first important aspect to understand and know more about it is to grow these organisms. Yes, if I have to work on one particular organism, I have to grow it. And how do I grow it? Now, I can grow them in certain cultures which is called as the media. Okay, media is actually called as the medium on which it grows. Now basically, uh, if I'm talking about a parasite, it would go into my body, consume my body part as the medium, gets nutrition, gets energy, and starts growing. Whereas there are other types of medium. If it is not parasitic, then I have to give it some kind of food which allows it to grow further. Now that kind of substance which allows the growth of microorganism is called as a medium, and when I make it, we call it as the media. Now this medium will have all the nutrients for the growth of microorganisms. It will have carbohydrates, the proteins, the amino acids, the hormones, everything it has. And hence, we call this media as the nutritive media. We call that as the nutritive media. Now, what I generally do is I take this nutritive media and I put it in a Petri plate. Okay, or what I would do is I would put it in a conical flask. I would put it in a Petri plate, or I would put it in a conical flask. So here I would put it here, and then I would spread the nutrient medium here. And then what I would do is I would take a very small amount of organism. Okay, maybe some very small amount which I can't even see from my naked eye, and place it on this nutritive media. Now this small lump of organism that I have taken gets all the nutrition from the media and starts growing, and it would form a huge big clump, and this big clump can be seen from our naked eyes. And it is because of this kind of media that we are able to do lots of work in the case of microbiology, and also using microorganisms for various purposes. In fact, right now we are using microorganisms to produce insulin, and what we do is we are taking the organism, growing them in this kind of conical flask, and take insulin out. So this is what we keep doing. So here you see that the growth of the microorganism on a nutritive media is something that is very, very important. And one thing that we should always remember is that when I'm growing this media, organism on the media, I have to see that there is no contamination. I do not want all the organisms to grow in that place. I want only the organism of my interest to grow in that area, and that is what we aim at in our microbiological laboratories. Now, once I start growing the organism here, then I'm going to use it for various purposes. So I can use these organisms to treat certain diseases, or to treat certain or to get some pharmaceuticals like insulin, or certain medicines like certain antibiotics, and so on. And that way I'm going to use these microorganisms which are going to help us to have a very good human welfare, right? So now, with this background, let us try to find out where these microorganisms are being used uh, under various uh, techniques, and how these microorganisms are being helpful to us from a very, very long time. So before we start off with anything else, I would start off with the use of microorganisms in our houses. Okay, so let us start off with the use of microorganisms in some of our household products. Now, do you think we have the use of microorganisms in any of our household products? Oh, of course. Uh, I'm sure we all of us eat curds, we eat buttermilk, or we have idlis and dosas for our breakfast. Now, all this have microorganisms in it. Okay. Uh, in fact, we use many microbes and many microbial products in our everyday life. Okay. So I was telling you that we eat curds. Where do we get curds from? Very much right. We get it from our milk. So what we do is we take milk, and if you know how we do curds at home, we take a very small spoon of curd and put it into the milk. We call that as a starter culture. A very small spoon of curd would have certain microorganisms which we call as the LAB, standing for lactic acid bacteria, or we have one of them which is called as the lactobacillus. Okay, we call that as the lactobacillus. So we have the lactobacillus or the lactic acid bacteria. Now this one spoon of curd would have all these organisms, labs. And when I put this starter culture into the milk, you see over a period of time, say close to around uh, 6 to 8 hours, depending on various other conditions, you see that the lactic acid bacteria would produce certain acids which secretes out, and that gets coagulated, and then it partially digests the milk protein. Okay, uh, this uh, you know digestion of milk protein, partial digestion of milk protein, and the coagulation will convert milk to curd. Okay, but though we say that the starter culture is very important, you should also know what affects this. You might have seen that before we make curd at home, we will slightly heat the milk. Okay, it should not be boiling hot, but it should be somewhere around 40 degrees. Now, when we heat it at around 40 degrees, you see that this activates the growth of lactobacillus and labs in our starter culture, and they start growing very, very fast in the first few hours. Okay, so this kind of uh, triggers them to divide and is very, very important for converting the milk to curd. Please note that curd is very, very important for us. You know why? Because curd has this organisms, right? And this organisms also increases vitamin B12, a very, very important vitamin B, which is very important for carrying out various functions in our body, giving us energy. It is a kind of it is a very important vitamin which helps to keep many of the enzymes active. So this lactobacillus gives us vitamin B12 as well. Okay, also, if there are any kind of disease causing microorganisms, this lactoacid bacillus will overpower them, and this grows faster than the disease causing microorganism, and therefore it checks the growth of disease causing microorganisms. So this way you see our microorganisms are present in curds, very, very important ones. Then I will give you one more example of eating our breakfast like dosas and idlis. Dosas and idlis are considered to be very staple breakfast for South Indians. We always like lots and lots of dosas and idlis. Every day we can eat dosas and idlis, right? And if you have noticed how they make dosa batter or idli batter, you have a batter, right? Now, the batter that we make here is also very, very specific. You know how we do this? Uh, what we do is we take rice and we take some kind of proteins. Generally, we take urad dal. So we take this kind of dals, and then what we do is we kind of mix them. The next day morning, you would have seen that the entire batter would have puffed up. Okay, it would have puffed up. And then you take a ladle and you mix it well. You can see some small bubbles coming out, which is nothing but carbon dioxide. Okay, now you see that overnight, if you leave that batter, there is evolution of carbon dioxide. Okay, so where does carbon dioxide come from? All this happens because of the glycolysis that we spoke of. Okay, uh, anaerobic digestion of starch or glucose gives us carbon dioxide, and thus you see we can make batter which is very, very good. Not only that, you also have the pizza dough and the bread dough. Even there you can see that small uh, air bubbles coming out. Your bread becomes very soft only because of the carbon dioxide. Now, which organism are we going to add here? I didn't add any starter culture. You remember when I'm making dosa batter, I didn't add any dosa culture, starter culture there. But overnight, you see that it grows faster. Now, how does that come from? Now, that comes from some of the organisms that's present in the ladle, in our environment, and in our hands. You might have seen that people say that in somebody's hand the dosa is very tasty. They mix it like in their hands, and you see that it comes out very well. Why does that happen? That happens mainly because of some of the organism that's present in our hand going into the batter. And same thing even with making the dough for bread also. But generally, when we are making any kind of bakery products, we add something called as the baker's yeast, or that powdered yeast that we get. That powdered yeast is added into the dough, and it is mixed well. You keep it for around 2-3 hours, and you see that the dough actually increases in size. They generally cover it with a warm cloth or plastic, and once that puffs up, It is used to make bread and uh, some kind of dosa, or not dosa, sorry, what is that called? Pizza and burger bases. Okay, so like this, we have microorganisms used in our food quite commonly. Now, there is one more, I don't know if you have heard of this. There is one drink in our uh, thing which is called as toddy. It's a traditional drink generally made in this ghat section of our South India, where there is of sap, plant sap, and that plant sap ferments. Okay, it's from a plant, a palm plant. The sap is taken and it is fermented, and that gives you some kind of alcoholic drink, which is being used quite commonly. Along with that, we have various types of fermented foods. So we have fermented foods like we have fish. Fermented fish is something which is very commonly used. Then we have fermented soya bean. Then we have fermented soya bean, and then we have bamboo shoots. They make pickle out of this fermented bamboo shoots. Uh, there is bamboo shoots there, which can also be used as food. And then we also have many vegetables which are kept in some kind of fermented products, and all these are being used as pickles or appetizers. Anyway, so we have all this happening mainly because of certain microorganisms. Okay, we forgot about one more important food which involves microorganism, which is nothing but cheese. One of the oldest food for which microorganisms were very widely used. These cheeses, I don't know the names properly, but I'm sure you would know. You have something called as Swiss cheese and various types of cheese. All these cheeses, they vary in their texture, they vary in their flavor, taste, based on the type of microorganisms that we have used. Okay, we have something called as cheddar cheese and so on. Okay, so all this is because, I mean, they all vary because of the microorganisms that are employed. Okay, there is one which is called as Swiss cheese. Now for that we use an organism which is called as Propionibacterium. Okay, and for others we have different types of microorganisms. There are certain types of cheese on which certain fungi grows on them. So this way you have different types of cheeses, and each one of them vary in their flavors mainly because of the microorganisms that are present. In fact, in our kitchen, I don't think we can live without having microbes around. Even otherwise, microorganisms grow very, very effectively in our kitchen, mainly because we have lots of food materials. And these microorganisms, many of them are good, some of them are bad. So as people who work in kitchen, should be quite aware of which one is good, which one is bad, and and uh, use it as effectively as possible. Okay, so this is all about our microorganisms in household products. The use of microorganisms that started long time back, almost at the time when we started with our agriculture. So that was a time when we started use of microorganisms as well, of course by accident, and then we try to improvise on these techniques. Now once we know that microorganisms are so powerful and so much useful in our life, why don't we apply it to many industrial products? It has to be, right? So our next topic that we will study is the use of microorganisms in industrial products. Now when we are very sure that microorganisms, many of them are very good to us, we realize that microorganisms can be used to make certain valuable products. And these valuable products obviously should be important to us, should be helpful to us. When I say us, it is very specifically to human beings. And uh, we use it for various purposes. Now, when we are talking about the use of microorganisms at the industrial level, the first thing that is very important to us is growing these microorganisms in a very, very large scale. I told you that I can grow the microorganisms on a petri plate or in a conical flask. But imagine if I want to have an industrial product. Can I grow it in a very small petri plate or even a conical flask? Now that is not possible at all, right? Now, when I am trying to grow them in large scale, I am going to use a large kind of cylinder-like structures, and that large cylinder-like structures, we call them as fermenters. We call them as fermenters. Now, what exactly is the role of these fermenters? Let me tell you how big this would be. Uh, Last year I was telling you that we have large, huge tanks which has the ability to hold around 1000, 5000, sometimes even 10,000 liters of liquid. Now what we do is we take such huge tanks and put in the nutrient media. Now obviously when I take 10,000 liters or even 5,000 liters, if I am not able to mix it well, all the microorganisms or the nutrients would be at the bottom, and then there would be some kind of waste growth that would happen. Now I don't want all that. So what we do is there is some kind of agitator inside this huge tank. Agitator will mix all the nutrient media and the microorganisms very effectively. Now when you keep on doing that, maybe after a day or two or sometimes a week, you see that there is enough microorganisms that is produced inside these fermenters. Now once the incubation period is over, I collect the liquid. Then if I need to take out the alcohol, I would take out the alcohol. If I need to take out any of the products from these organisms, I would take them out, purify them and reuse it. Some small part of this organism is kept safely and is used again as a starter culture. So this way I have starter culture also. At the same time I have growth of microorganisms also. Now most of our industries have large fermenters. The range starts from somewhere around 2 liters fermenters to around 20,000 liters fermenters. So we have this fermenters which has agitators. And now using fermenters, what all we can do? The first thing that we can do is obviously growing microorganisms. But then why should we grow them, and how do we grow them is the biggest question. Now fermenters are used for most of the industrial products, but however we would try to concentrate on two important products under this heading. The first one is something that we call as fermented beverages, and the second one that we would talk under this heading is antibiotics. When we talk about microorganisms, we call this huge tanks as fermenters mainly because microorganisms are very effective in bringing about fermentation process. And I'm sure you know what fermentation actually means. So like yeast, we have certain microorganisms which are being used to make wine, beer, whiskey, brandy, rum, and many such drinks. Now, for all these organisms, for all these products to be present, to grow them, what we need is we need a organism, and that organism is Saccharomyces cerevisiae, which is nothing but yeast. Okay, so we call it as brewer's yeast. This is the one which we make bread with, cereals and fruit juices and many others. Now it is also the same organism with which we make alcohol. Ethanol that you use in your labs is made by this organism. Okay, so we have glycolysis under anaerobic conditions uh, that would give ethanol, and this ethanol is being used for various purposes. Now, what kind of raw material I use and what kind of fermentation happens, okay, and what kind of processing I do, I can get any type of alcoholic drink. Say, for example, if I'm talking about wine or beer, you see that these are using Saccharomyces cerevisiae, then they are produced. Now they are processed without distillation. There is no distillation happening. Whereas if I do distillation, then it becomes whiskey or brandy. That is because of the distillation of the fermented broth. And also, not only the distillation part here, we should also find out what is the raw material. So if I use grapes, I get grape wine. If we use apple, that's a different type of wine. So we have different types of raw materials, different types of processing that separates wine, beer, whiskey, brandy, rum, and all of them. And uh, you see that each one of them is quite important for uh, maybe a recreational purpose. So this is all about the fermented beverages. Now the second thing that we would discuss is about our antibiotics. Heard of antibiotics? I'm sure you would have studied about the famous story of Alexander Fleming. Uh, so when we're talking about antibiotic, you know, antibiotic basically is derived from three words. The first word is anti. Anti is what? Against. Right? Anti is against. And what is bio? Bio is life. So basically, a chemical which is against life is what we call as antibiotics. Uh, so what exactly do these do? Now these chemicals, antibiotics, they are those chemicals which has the ability to kill life. And when I say kill life, we are specifically meaning with the microorganisms. Okay, some of the microorganisms which have entered into our body as a parasite, now they will be killed by the use of antibiotics. Sometimes they might not kill, but they would not allow them to grow. We call them as biostatics. Okay, so they do not help in killing the organism, but they will slow down the growth of those disease causing microorganisms. Hence, when we first talk about antibiotics, we should know that these are those chemicals which are inhibiting life, but they are helping us to kill the pathogen and thus helping us to save our life, right? Now, this was first discovered by Alexander Fleming as an accident. He was working on one organism which was called as staphylococci. Now, this staphylococci, he was growing, and because of some reason he could not wash the culture, and he could kind of see that the presence of some chemical stopped the growth of staphylococci in his petri plate. Then he was thinking, why is my staphylococci not growing? Something should be there in this. And then he found out that there was one fungi growing there, and that fungi was Penicillium notatum. When he saw Penicillium notatum and he saw staphylococci, he realized that somehow this Penicillium notatum is not allowing the staphylococci to grow. Now when this was happening, then it was like, okay, so what next and why it is happening? Then he realized that this Penicillium notatum was secreting one particular chemical which is called as the penicillin, okay, a chemical which Alexander Fleming named it as penicillin. Now, because of the presence of penicillin, staphylococci was not growing, and thus uh, the first antibiotic, penicillin, was discovered. And later there were many other scientists like Ernst Chain and Howard Florey, and they were kind of trying to understand more about this particular method of treating the infections, uh, especially during World War II, and they were, I think, awarded a Nobel Prize for uh, medicine in 1945 or so. Now, after penicillin, we have many other microorganisms which are used to produce lots and lots of antibiotics. Okay, so you have ampicillin, many others. All of them are being grown inside the labs, and their uh, their antibiotics are being purified, and they are being used to treat various diseases. Some of the diseases, deadly diseases like plague, then we have whooping cough, heard of flu whooping cough, and then the diphtheria, then leprosy. All this are being kind of controlled by using this kind of antibiotics. And even now, if you go to a doctor, even for a simple fever, he would suggest the antibiotics, because antibiotics are very, very effective. Since we're talking about the plus points of antibiotics, I would not go in detail with respect to immunology and tell about the negative aspects of antibiotics. Nevertheless, overdose or not a correct dose of antibiotics is not good for any person. So just remember that when you are consuming antibiotics, you complete the dosage for 5 days, 3 days, or whatever, and only then you would be stopping them. Okay, this is one thing that I would like to tell you. Also, I would like to tell you one more important thing here. Antibiotics comes from various types of organisms like fungi and very few bacteria also. But right now we have something called as the synthetic antibiotics. Now, what are synthetic antibiotics? These are the antibiotics which uh, are made inside the lab. We make some kind of chemical reactions such that we obtain the structure of those uh, antibiotics, and these are being used. So we have many natural antibiotics, and we also have various, uh, non uh, natural, which means to say a synthetic antibiotics present in us. So this is about the microorganisms that are used in industrial products. So we have fermenters that are being used here to make beverages and antibiotics. I would just like to add here, there are many other important things that we are going to make use of here in these fermenters. Say for example, I would like to make certain particular chemical, certain proteins, certain amino acids. All these are also, we use a lot of fermenters. Nevertheless, you would know that it's not an industrial product as such. We use it in various other uh, industries as raw material. So microorganisms as such are used in various industries right now, and we are happy using them in our industrial products. So where else we are going to use them is our topic for our next class. I hope you do not have any doubts. So see you in the next class. Bye then.